Support Microbe TV. It's time for our annual fundraising drive. We depend on your support to produce high-quality science videos and podcasts, and now is the time to help us. If our programs have appealed to your science interests, or if they have helped you in some way, please record a brief audio or video with your phone and tell us about it. Let us know how we empower your inner scientist. We'll use it for our fundraising efforts. Send it to incubator@microbe.tv. And for more information on how you can give us your support, go to microbe.tv/contribute.
From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on January 3rd, 2025. Happy Microbial New Year. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 73rd episode of Matters Microbial. With the new year, I have the work of many microbial centrists to share with listeners and viewers in future episodes. 2025 will definitely display both OMG, overwhelming microbial greatness, and WTM, what the microbe? This I can promise. Stephen Ripple wrote to me via the website and asked if we could have an expert on using UVC lights to reduce viral and bacterial contamination in hospitals. Thank you for the suggestion, Stephen, and I will look. Yesterday was the official grade submission deadline for the fall semester here, so I will not be teaching again until the fall. The last time I will be teaching microbiology at the University of Puget Sound, and I have all the feels. Last episode, I shared some Thank a Microbe artwork that my Bio 350 Micronauts made. Here's another about Wolbachia from one of my microbiology students. This is a fabulous approach to get students to feel ownership and energy about their learning. However, I need to be clear that the originator of this fun exercise is my friend and colleague, Dr. Ann Estes of Townsend University. I'll put her contacts in the show notes. Ann is a wonderful source of microbial inspiration for the community. The Quality Quorum. On my microbiology final exam, I had students investigate the relatively new concept of the holobiont, recently reviewed by Seth Bordenstein and many colleagues. Article is linked in the show notes. As you can see, one of my Micronauts came up with a fun meme to illustrate what they thought of the concept. I also had my Micronauts write haikus about microbiology. Like this one about retroviruses. Retroviruses, moving backwards through genomes, like time travelers. It's pretty awesome. Or this one, describing the Micronauts' perspective on the microbial world. Observe microscope. Tiny life forms appear now. Do they see me too? I actually wrote, when I was correcting it, yes, they do. Again, I have long believed that using creative approaches in the classroom pays off for both instructor and student. Try it yourself. The enthusiasm and creativity of my students really energizes me. It has truly been a privilege. During several episodes of this podcast, we have discussed the gut microbiome. The way in which it's studied seems almost remote, where there's lots and lots of short reads and assembles and metagenomes, and all of that is wonderful. But what if we could see the dynamics of the gut microbiota in real life? Well, we can. Thus, I am beyond excited to have Dr. Travis Wiles of UC Irvine on the podcast to discuss the research his team has done doing exactly that. Welcome to the Quality Quorum today, Travis.
Thanks for having me. It's great to be here.
I especially appreciate your willingness to be the first episode in 2025.
Hey, someone has to do it.
Yeah, I never believed that I would live this long, to be honest, but there you go. So I think this is a wonderful idea. We've talked about the microbiome in several episodes, and one of the things about it is how it's studied. And people have talked about the populations that are there being seen through 16S ribosomal RNA, mostly through short reads. And lots of interesting things have been done, but not a lot about the dynamics of the microbiome. There have been lots of snapshots that have taken place over time, and it becomes very computer science intensive, which is not a bad thing. But you have a slightly different approach. What brought you to the microbiome?
Yeah, that's a big question. And I think to answer that, maybe I got to go back to when I started grad school. I actually got my training in bacterial pathogenesis, so studying the bad bugs, right? Trying to understand all the genes and what we call virulence factors, these sometimes toxins they secrete to manipulate their hosts. And I was really captivated by that. But, I think kind of like hearing your intro really resonated with me because when we talk about and think about microbes in general, we're all in our collective thought bubbles, individual thought bubbles. What are we imagining? Little cartoon characters that we find on YouTube, or maybe some one scanning electron micrograph that we saw one time. So I had this strong pull that I wanted to see these systems, much like a field biologist might be studying kind of a rainforest or an ocean, right? So you make observations with your eyes and you write them down and you make connections, you watch them in motion. And for me, very early on, before, I was really aware of microbiome, I was just really drawn to seeing microbes. And so I was drawn to approaches such as fluorescence microscopy in general, to just really see them, to kind of inform my intuition of how they were functioning. So that's really where it started in bacterial pathogenesis, and I applied those tricks there. But then right around 2009, 2010, when the microbiome was really kind of gaining momentum in its early phases, I just got really captivated by going from studying maybe like a single microbe, a pathogen and its host, to then thinking about these really complex communities that are interacting with each other, interacting with their hosts, and that just seemed very alluring to me. And so that's when I got into the microbiome field and really brought this kind of approach to studying microbes, live imaging, and things like this to studying the microbiome.
It's really interesting that you bring this up because what you can probably see now is a cartoon that Beatrice, the biologist, made for me some years ago. And for those of you who are just listening, it's really relevant because what you've got is a professor like me taking role in a class, and it's not just the students, but their microbiome and responding to it. So once again, you see mostly in class, it's the microbes, which is an interesting idea. Secondarily, the idea of looking at things on a fixed slide or through a microscope after fixation and all the rest has always troubled me. So fluorescence microscopy, especially of living cells, is particularly interesting. And for listeners and viewers, would you care to talk a little bit about fluorescence microscopy?
Yeah, right. So, microscopy in general has a lot of different uses, applications, and techniques. Fluorescence microscopy is an approach where we can take naturally fluorescent molecules, maybe engineered fluorescent proteins. I like to teach my general microbiology class about where some of those proteins come from because it's pretty cool. They come from nature, right? So there's organisms out in the environment. We've kind of combed the landscape, not me personally, but other researchers, chemical biologists have found these proteins being made by fish, jellyfish. It's really amazing. So we take those, we can clone those gene sequences, we can clone them and use them to engineer bacteria to then express them, and it illuminates their cells. And then you can use that in combination with techniques known as fluorescence microscopy to see them in their, some cases, they're kind of in situ or their native natural place of being.
It's such an interesting idea, and I think many people that have been following this program have seen some of the work that I've even shown using green fluorescence protein from a jellyfish as kind of a monitor of an organism being there, and sometimes even gene expression by that organism. So it's an extremely powerful tool, and once again, it takes you away from something that you alluded to before. If you were studying rainforests, and people brought you specimens individually to look at, it wouldn't tell you much about the interactions between things. And that's really hard. And I'm not denigrating early work at all. What I am saying is that we use the tools that we have available, and as tools improve, so should our approaches. For example, people studying biofilms, and they would study one bacterial species in a biofilm context, and that's not how they are in nature. And it's funny because I once said that many years to someone, and they said, well, yeah, we're working on 2 or 3 together. And I just did this because, in fact, that's also not how they are. I mean, we can make a comment that the gut microbiome, certainly of people, is as complex as any rainforest you've ever heard of. So how do you study that level of interaction between things? And that's what really drew me to your work.
Yeah, I mean, I had a friend who's also does a lot of live imaging. I remember one time he was kind of describing his kind of view and his relationship to his work, and he showed me this, it kind of reminded me of, like these little cartoon flip books, right? Where you have kind of these animated pages, and then, you kind of flip through the pages and you see like a horse running or something. But if you stop on any one page and you say, okay, tell me what's going to happen in this story based on this one image. Well, if you happen to land on a page, say, if it was a horse running where the horse's legs were off the ground, and let's say you didn't even know it was a horse, you're just like from another planet, you're trying to understand this system, you'd be like, oh, these interesting animals hover above the ground, or you just have no idea how to connect these. And so live imaging, fluorescence microscopy, you're 100% right. Like kind of the single time point static imaging certainly gave us a lot of clues. It kind of gave us the landmarks, the lay of the land. But something you're confronted with as soon as you start doing live imaging is that there's also not just this spatial scale that now you become aware of. There's also this temporal scale. This is how things change in time. And it really kind of brings up a lot of new questions about kind of the dynamics, the kinetics, how fast do these interactions, relationships form and break, and things of this nature. So yeah, I think exploring the microbial world, through 4K Technicolor vision is really fascinating. Kind of starts bringing up and conjuring questions we didn't even know we knew to ask.
And it's funny too, because it's very easy to be misunderstood. And I don't want to repeat myself on this, but I don't want anyone to think, and you certainly made this point as well, that there's any criticism of earlier approaches. Here's an example. A lot of human microbiome work is done by looking at fecal samples. And I've, for many years, said that I don't know that that really represents what's on or around the gut epithelia. Because at the sake of being rude, you've got this giant pellet, for lack of a better term. Mm-hmm. And the inside of that pellet is going to be very different from the outside of the pellet. And all of that's going to change over time. And when I've had that conversation with people, I had to be very, very careful not to criticize the approach in the past, because people become very invested in their work. But just as you said, we need to be open to new approaches and new perspectives on this. And as you say, the temporal business is critically important. And you can do that with your system, which is wonderful. And that brings me to what model system that you think that you do use.
Yeah, well, we use the Zebrafish, and actually, Mark, I'm gonna kind of, maybe for your listeners, your viewers, kind of follow up on this kind of thinking about the concert of model systems and approaches that we all use. Kind of as you were describing that, for me, I try to tap into my colleagues who don't do live imaging, but do the sequencing and do all these other descriptional techniques, or maybe like deep cell and molecular biology, chemical biology, because to me that provides this raw material, this motivation to say, I want to see that. I want to see the spatial, architecture of how those interactions are occurring, and something that other people might find useful, thinking about how we all kind of use these disciplines and kind of our individual superpowers. There's this parable of the blind monks, right? Where all these blind monks are trying to, they're kind of, I think they have their hands on an elephant, and all of them are describing what they're sensing and feeling. And someone's like describing the tusk, and someone has the tail, and someone has the ear, right? And they're all describing completely different things, but it's all part of the same truth, right? They're all they're all describing an elephant. And so I really kind of view my work and the work of my colleagues in the field in this way. And it becomes really enjoyable in that sense.
I agree. And it's really true on every level. I mean, most of us, we get used to doing things in a particular way, and we lose the excitement of bringing in new ideas. And as you just said, when you have folks who aren't trained in that area, they ask the best questions. They have the best points of view. That really inform the work of many other people.
100% agree. 100% agree. Yeah. So let's, so zebrafish. Yeah. So our window to the microbial world is actually through this animal, this zebrafish. Right. And so the zebrafish is actually this pet shop fish. You can go to your local pet store and find one perhaps. What makes them great pets is that they're really easy to raise and grow. They're really small, only a couple inches long, which is great for us as scientists, because then it means animal care is easy. But the zebrafish historically it's a powerhouse model for studying genetics and development. First vertebrate that was cloned, for example. But why it's such a powerhouse for development is because of its right. So it's a fish. They lay eggs, and the eggs are fertilized ex utero, and you can literally watch this single cell. Well, at that point, this single cell grow and divide. Really in the course of 24 hours, you go from a single cell to an animal with a beating heart, a brain. Blood flow, 2-3 days in, you got a working digestive tract. You can all see this happening. And so this is why zebrafish power developmental biology. But then there's a handful of people, who have come before me, Karen Gilman, John Rawls, who started, using zebrafish, larval zebrafish in particular, because their bodies are transparent, right? So we can use microscopy techniques to see inside of their tissues and their organs, especially the gut. And so we can use this animal to start getting clues onto some of these things we're talking about, these spatial and temporal dynamics of interactions between the microbes and, of course, with the animal host.
And it's really amazing, because I'm thinking transparent, and then I'm thinking fluorescence, and there you go, right?
That's right. Yeah. So when you combine the two, because microbes, on that scale, there are microbes that make pigments, but essentially when you combine them inside the gut with the animal tissues, everything just kind of looks the same, at least based on light microscopy, right? So the light's bouncing and refracting, and really just can't make sense of it. But then when you illuminate certain components of the system with fluorescence, so you genetically engineer the bacteria, what's cool about zebrafish is we also have a lot of what we call transgenic zebrafish that have been engineered to express fluorescent proteins at different parts of their tissues. So now you illuminate not just the microbial world that's living within their digestive tracts, but then you illuminate the tissues and the biology. And so really you start to have a chance to observe all these systems interacting with each other. We can go in a lab. We have several microscopes in lab, and what I love to do is bring people in, whether it's a visiting professor, an undergrad, and I love to show them zebrafish. One of the things I love to do is get like a 2-3 day old zebrafish, right, before they start developing their pigments, and you can literally kind of put the person on the microscope, and then their eyes, kind of light up, and it's almost like I don't even know what I'm looking at here, but like intuitively they do. They're looking at a vertebrate animal's biology from the inside out. They can see the blood flowing, the gut kind of wiggling, right? These are all things we intuitively know, but then when you see it, I think it just really kind of captivates you. And there's often a period of silence as people are trying to take it in and trying to understand what they're seeing. And of course, we have, then when we, then see microbes, bacteria inside of the gut, I think then people, and I still get this even when I present this at, to my classes or at seminars, I show people bacteria inside a vertebrate gut. And it's interesting how people kind of approach that. Sometimes they're like, wow, that's amazing. Never thought about it, never seen it. But then some people kind of come to it being like, well, why is it like that? Or why? They're kind of a little bit resistant. And that kind of goes back to what I was saying before. I think we've all formed these opinions and thought bubbles based on these cartoon diagrams in our head. And when we see the reality of it, I think we really do have to go through an adjustment. And that's actually what I find really compelling and fun about this type of work. It's super challenging. I feel like we are looking out. I mean, if we scale up and think about, astrophysicists who, turn the James Webb Telescope out to the cosmos, we've never seen these things before. How are we supposed to make sense of them? Could we have even predicted what we're about to see? Yeah, so I think this is incredibly fun.
I know, and the whole concept, I mean, most of us that have been at one point or the other a biology major, when we're taking developmental biology, or in my case, cell biology, and we would look at, say, a chicken embryo, we'd be able to see the blood moving in the embryo. And all of that worked out really well. But what's interesting is that's not a manipulable, at least yet, manipulable kind of system. But it is manipulable on both sides. Over and over again, we've seen that model systems involving, for lack of a better terms, microbes and macrobes, for lack of a better term.
Yes.
The best systems are ones where you can manipulate or analyze both sides of that equation.
Yes. Yeah.
And you've got one. And zebrafish are pretty remarkable. I was an aquarist as I grew up, and they were the one type of fish that I could keep without killing.
Yeah. Well, that's what I'm saying. They're easy to raise, right? Yeah. And what's cool about them too is there's, people, I would say, well, and I forgot to mention Lali Ramakrishnan, who works on Mycobacterium marinum, tuberculosis relative in zebrafish. Lali Ramakrishnan's lab, John Rawls' lab, Karen Gilman's lab, they Judith Eisen make tremendous use of the genetic amenability of zebrafish in their work. I would say for us, we too are as well, right? We use animals that say don't have an enteric nervous system, so their guts don't move quite right. So then we can ask these questions. Okay, if we think spatial structure is so important, let's turn off all the systems that make the gut do these peristaltic motions, right? Kind of jumble up everything. Let's turn those off and then see what happens. And so, but other ways in which zebrafish are amenable because of their small size and they're kind of sturdy, right? Like you can house them in many different configurations. And so I guess the first thing is we can raise them germ-free, right? And this is actually quite easy compared to like mouse models and other kind of vertebrate models, right? So again, back to these eggs that are developing outside the body, we can actually take these, we just like wash them with a little bit of bleach and iodine, put them in some sterile water, and then we just divvy them up in the standard tissue culture flasks, just like you would in tissue culture labs. And then you just maintain those and then you can raise those for several days, up to a week, germ-free. The other thing that we've been doing, because they're so small, larvae are only about like 5 millimeters big. They're actually, when you compare like a larval zebrafish to say like Drosophila, a fruit fly, you're pretty surprised that the fruit fly is actually quite big compared to a tiny larval zebrafish. But what this means is we can actually put larval zebrafish in little multi-well plates, and we can ask questions about, well, if we just raise an animal by itself in isolation, essentially quarantine, how does its microbiome assemble? What are some of its functions? How do those contrast compared to when we raise them in large groups where microbial transmission is affecting it? And some of my colleagues back at Oregon, Brendan Bohannon's lab, along with Karen Gilman's lab, have shown these connections that yes, certainly it does. affect microbiome assembly, and we're carrying on some of that work ourselves. So yeah, so zebrafish are really amenable in many different ways, not just genetically, but just like the way you can house them and arrange them.
So two things really quickly. The first is many people, especially I call them new micronauts, people that maybe don't have a lot of experience in microbiology yet. We hope to change that.
Yes.
Have actually seen zebrafish that have been genetically engineered in aquarium stores.
Mm-hmm.
And they're often called, I don't like this term, but they still call them that, glowfish, trademark.
Yes.
And all that is the GFP and RFP and similar types of fluorescent proteins, I believe yellow as well, have been inserted into them. There's a long story behind that I'll put in the show notes. But the point is, people had been genetically manipulating zebrafish for a long time.
That's right.
And it's wonderful. Secondly, the issue of how microbiomes are transmitted between generations is a really interesting one. For example, humans obtain it through passing through the vaginal tract and often through maternal care. But I'm presuming that the fish are getting this from the environment.
That's right. Well, I mean, this is also, kind of a complex question. We wrestle with this a lot. And what is environmental? What is direct host, maternal to, offspring transmission? As I said, we're doing some of these experiments. And so we kind of wrestle with this question of microbiome inheritance. So if we have adult animals that, spawn, lay eggs, and we can certainly collect fecal matter from the adults, and we can kind of see what microbes are there. And then we do this thing where we take all their babies, essentially their larval fish, and we raise them in different configurations. And yes, I would say generally this is right that they're inheriting it from the environment, but there is a strong influence of the parents. But that will come down to like the proximity of the parents. Like some organisms in the environment actually deposit, microbial communities onto their offspring. The zebrafish seem to lay them and they just go about their business. They're as likely to eat their own eggs as they are
Yes.
So, yeah.
You know what else is interesting about this is that whenever I talk to people about science in general and we talk about model systems, people who aren't biologists will sometimes critique the use of, for example, the roundworm Caenorhabditis in developmental biology, saying, oh, that has nothing to do with people. Or Drosophila, that has nothing to do with people. And it makes me, shake my head and everything. But the fact of the matter is, zebrafish are a really good system because we're used to the idea of fish. And I know for a fact that you can train fish to come to the top of the tank when you tap because I did it.
Yes, it's true.
So all I'm saying is here's a model system that is sufficiently complex that I think average people can connect with it.
Yeah, 100%. I mean, there's a temptation, I think even the way when we think about the microbiome, of course, from our human-centric point of view, and I know we're microbe-centric here, but from a human-centric point of view, I think people they see these model systems. And so maybe I'll just say, we need model systems. There's no way we're going to crack the code on symbiotic relationships without the use of model systems. But also each model system tells us part of the whole truth, right? So, but what I would say about zebrafish is, and kind of when we approach it from this human-centric view, is it's tempting to always want to relate it back to human health. And this is justified. It's where a lot of the federal money goes. We want to solve societal issues around health. But the reality is like we really care about environmental systems and environmental microbiomes. And so fish are something that agriculturally is important to us. They're pets. They are indicators of the health of ecosystems out in, fresh and saltwater areas. So, something I always like to approach our work with is it's a really flexible, agile system that we can think about it through all these different lenses. And I think this is really important. And also, really fun.
Yeah. So what kinds of questions are you looking at in your lab group that involve this wonderful system?
Yeah. So I would say, the questions are really emerge from this huge change in the way we view the microbial world. As I mentioned, I got my start studying bacterial pathogenesis. So there the goal is to focus in on a particular species or strain of a bad bug that causes a disease. And we got to find ways to eradicate it, get rid of it. We want to stop it from being in our bodies, stop it from causing disease. But then, the microbiome kind of era has ushered in this new view of the microbial world. And that's one where we don't want to eradicate microbes. We don't want to stop them from getting us. We actually want to cultivate them. We want to nurture our kind of internal ecosystems by adding in, say, like fermented foods and stuff like this, right? So this is a huge paradigm shift in the way we think about microbes. And so this has given rise to this idea of, we can solve, societal and health issues by engineering microbiomes, by building microbiomes. And so this is a large idea that motivates the questions that we have. And we want to figure out, if you're going to understand how to build a microbiome, where do you start, and how do you kind of brick by brick kind of assemble this broader picture of microbiome? So questions that we're interested in is, how do microbiomes assemble right from the single cell, like thinking about single cell, biology, bacteria, how they get into, say, a digestive tract, and how they respond to that environment, how they start augmenting their interactions, and kind of, if they're pathogenic or going to cause disease, or if they're beneficial, like what dictates whether or not they're going to be turning on the genes that kind of are mutualistic versus parasitic, and things like this. So we're really interested in that interplay, but we also kind of apply this thinking and the approach to thinking about, really relevant things like antibiotic resistance. Yes. I think that the assembly of a microbiome, right? Because antibiotic resistance is often, arises because bacteria, they like to share genes, right? They can do this really amazing thing where they can take, a bug can inherit genes horizontally from its, neighbor. And so often they'll share resistance plasmids and even transposons via those plasmids in that way. And so when you think about, bacteria inheriting DNA and sharing resistance genes, this becomes a spatial temporal problem. And so this is one we can actually get a new look at by using live imaging. So we're really curious about how the spatial configuration, the temporal dynamics inside the zebrafish gut actually fosters or maybe inhibits, cuts off those chains of transmission of these plasmids and resistance genes, stuff like this. So I would say that's one of the kind of big areas right now, is studying antibiotic resistance, but also just more generally mobile genetic elements, including bacteriophage.
Well, see, this is interesting because one of the things that you said, and I want to emphasize this for listeners and viewers, is live imaging. Because most of the time when you're looking at these things, we are removing nucleic acids from organisms. So we've killed them to remove those nucleic acids. It's a snapshot. And yes, if you do a sufficient number of snapshots, you can recreate something. But this is different because the system is alive. And as it changes through environment and things that come in and leave, you get to observe that in situ as it happens. And that's really exciting.
That's right. Yeah. I would say, so even before, taking on some of the projects that we have going in lab, we would sit around the table and think about, this must be the case. Everything you just said must be the case, that snapshots are not telling us the whole picture if we only had the dynamics. But even at that point, before we did the experiments, I don't think we would bet the farm on that we knew what was about to happen.
Right.
And maybe kind of a good kind of illustration of this is this recent project that we had going in lab. We kind of affectionately call it the Follow Project. It's a project in which we took on the challenge of going from seeing the microscopic world within, like, say, a living animal's gut microbiome, right? The bacterial cells, those live on the micron scale, right? But through the years, as I've been doing live imaging in zebrafish, I thought it was possible that we could actually image phages, bacteriophages, bacterial viruses, right? Which live on the nanoscale, right? So for scale, typically a bacterium is usually 1 micron long, right? 1,000 nanometers, maybe 2 microns long. Phages are typically, They come in many different forms, but a very common one is they kind of look like this lunar lander structure. They have this capsid that's often between 60 to 80 nanometers. There you go. Perfect. Yes. That looks like a T4, T3 style phage. Yeah, so those are these kind of myoviridae, these tailed phages, right? And then they have these kind of tails that are roughly 100, 150 nanometers long. So in all, they're about 150, 200 nanometers tall, right? So they live on the nanoscale. But I already brought up mobile genetic elements, and when you look at the genomes of bacteria, they were forged by phage. I mean, phage have been jumping in and out of those genomes, rearranging those genomes, bringing in new genes. Again, back in bacterial pathogenesis, to realize some of the baddest bugs in history, Shigella, cholera, salmonella, their virulence factors, the genes they carry to make them what they are, were carried in by phages. Phages, the movement of phages created those bugs. And they're also creating good bugs too. I think we know less about that, but I'm sure they are. So this is what leads to this motivation to say, we got to see what the phages are doing. We have to see it, right? We have to see it go, kind of in addition to not just sequencing them. So anyways, the Follow project. So kind of the name Follow, kind of it's in the name, right? We wanted to follow the replication and transmission of bacterial viruses. And so what we did is we engineered viruses to contain these little protein tags on their capsid surface. And then what we do is we express a fluorescent protein that can then bind to those capsid tags. And so what ends up happening is you have this situation where you have these bacterial cells and they're fluorescent, but they have the ability, once a phage starts replicating within them, and as those capsids become formed, the fluorescent protein, the tag, starts relocating onto those capsids, and all of a sudden it comes into view. You can see these capsids forming inside of the cell. And what's amazing is that then you watch, so a lot of viruses, the end of their life cycle within their cell is they kill their host through kind of this, in some cases, what appears to be this explosive lysis that happens, which actually takes many different forms across the viral, the virosphere. But anyways, so we have these images where we were able to capture this process of viral replication, lytic replication, as we call it, happening inside of bacterial cells. And then if we use DNA dyes in the media that can't go inside the cell when the cell is alive and intact, but once the cell ruptures open and lyses open, the DNA is spilled out into the environment, and then the dye labels that. And so you can really get this dynamic view of what happened. So this was kind of our first, this was kind of the fun phase. Well, the whole Follow project was fun. But these early days of like, can we do this? Oh my gosh, we did do it. And then now employing this. So we really can see this replication and dynamic of phages inside cells using Follow.
It's really interesting because, yes, you have a beautiful logo for the Follow system. Yeah. There's a wonderful paper about it, and I'll put a link to the show notes, in the show notes about it. And it's interesting because I'm used to the idea of not being able to see things with a light microscope on the nanoscale, but you've got a workaround for that.
That's right. Well, so there's so much innovation going on in microscopy, and I'm a trained geneticist, so I'm not my background's not in physics, and so there's a lot of incredibly creative people who are putting to use kind of, physics and kind of knowledge of light dynamics and how it interacts with objects to create new microscopes. And so, oftentimes if you have a bright enough signal, right, you can detect very small things. Now you can't resolve it very well. Mm-hmm. So like you can see like a nanoscopic virus, it'll look like a little, a bunch of blurry fluorescent pixels, and at least you can detect it. But of course we have access here to super resolution microscopes. And, I have to say, so our Follow project, and this kind of hints at, some of the challenges. And I kind of stumbled a little bit before talking about how fun this project is, but it was incredibly challenging. And, something I had been, through my postdoc and training, I had been mulling over this idea of how we would tag phages. But I have to say, like, this project only came to fruition, came to life because of the people in my lab who are working on it. It took a lot of my assistant professor colleagues contributing resources to do some of these techniques, and took essentially my whole lab to kind of endeavor and take on the follow project. But I have to shout out to the first author of this paper, Liz Ortiz. Just her creativity and ingenuity are off the charts. She's really fun to work with. She's a disruptive scientist in terms of, really challenging me, challenging others, challenging herself. And so really it was her that really kind of, once we got the phages tagged and we could see it, she's the one that had the eyes and the microscope. She's the one that really kind of took it upon herself to figure out how do we actually use all these different microscopy techniques to see these phages, not just on a cover slip or like a glass slide, but inside a vertebrate animal. And that took an immense amount of creativity and also just stubbornness, just like not giving up on it.
And this is important for listeners and viewers. There are some people who are change agents, and maybe your colleague Liz is a change agent.
Yes.
In fact, you should get her a lapel pin that says that.
Yes. I mean, it's funny. I always viewed myself as a change agent, but I have to say, for your listeners and viewers, as an assistant professor, junior faculty member, learning how to not be the change agent up front, but kind of facilitating and identifying these change agents. Yes, Liz is definitely a change agent. Yes.
And it's interesting, too, because I just finished teaching a semester of microbiology, and we only have the one course. And one of the things I'm very careful about is talking about the role that phages have in bacterial evolution. And I mean, if you go looking in the genome of E. coli, there are at least 10 remains of defective prophages. So what that means is that virus integrated its DNA into the host chromosome. And since the hand of Darwin is on all that lives, mutations happen randomly, and those mutations that prevented this integrated bacteriophage from leaving stuck it there. So I call it a rusting evolutionary hulk. But the fact is, it's still additional genetic material on which natural selection can work.
Yeah.
In addition, the concept that you alluded to is phage conversion. The fact that many different bacteriophages can carry genes as hitchhikers that can benefit another bacterium, sometimes in ways that we would prefer than not. You mentioned virulence factors, for example. The botulinum toxin is carried by a bacteriophage.
That's right, yeah.
And that's just one off the top of my head. I think the same thing is true for tetaноspasmin, the toxin for tetanus. Yeah. So this is a very common horizontal gene transfer mechanism. So many times on this podcast, we've discussed the dynamics that have to be taking place in the microbiome. And you've added a level of wonderful complexity, which are these, for lack of a better term, I dislike calling bacteriophages predators because I love bacterial predators, and they're not cleaving ATP to do it, but we can call them predators or parasites. They definitely are impacting the kinds of populations that you see within the gut. And people hadn't thought about that. I have a former student who's working on that as a postdoctoral student. Fascinating stuff, untapped. There are so many unknown bacteriophages out there as well.
That's right. Yeah. I mean, I really enjoy, kind of my trajectory of thinking about pathogens and then thinking about beneficial bacteria, thinking about those dynamic fluid definitions that we have. It extends all the way down to the scale of a single bacteriophage and its bacterial host. They're also doing this dance of mutualism, parasitism. They're in it for themselves, and then sometimes they're in it together. And so this is really fun to think about. And this has also been something that as we develop the Follow project and we start seeing now for the first time, at least us personally, and a few other labs around the world have been looking at phage and by live imaging techniques, it really does start, like I was saying before, we could sit around a table and kind of predict what we're about to see. But until you see it and you see the dynamics, it starts, now all these new questions start coming in. You're like, hey, wait, I think this, to your point, they're not just predators. I think they're working together, right? So yeah, these things come up all the time.
You no doubt are very aware of the emerging field of sociovirology.
Yes.
And this is 100% involved in what you're talking about. We had a guest on the podcast talk about that recently.
Yeah. Yeah. 100%. I mean, I would say, once you start looking at populations of bacterial cells, with actively replicating phages, this is again, going back to what it means to inform your intuition visually by watching a system, right? Something that Liz often says when she's watching these viruses replicate in bacterial populations, she often has this feeling that they know who, how many of us are replicating and where we're going and don't infect me, like, things like this. So I think we see it. We're not studying explicitly right now, but we see this. We see I think the specter of this social interactions between phages.
I get accused all the time of inferring intent to the microbial world. And I do, but it's not a conscious thing, at least, well, privately, I suspect it, but that's me. The fact of the matter is there's so much complexity going on. So how do you tease that out? And that's the good news. Now you're able to watch particular bacteriophages as they interact with the population structure within this gut of a zebrafish as it's developing.
That's right. Yeah. No, I mean, 100%. We're now traversing into this iterative phase where now we've sat around the table, we've came up with ideas about what we thought we were going to see. Now we've seen some things, and now we can play this game. Let's play it from the bacterial side of view. Let's play it from the phage point of view. Let's play it, play it from the virus cell point of view, right? The fact that it's not one or the other, but the union between the two. Right. And so, yeah, so now we're in this iterative phase of kind of rearticulating our hypotheses and then setting up experiments to test those ideas.
So, Travis, several times in the podcast, we've talked about the rise of multiply resistant bacteria in different locations and situations. And what I'm trying to do is put a link to what you've been discussing with regard to bacteriophages moving things around as one route, but how that relates to temporal and spatial differences that you might see in the gut.
Yeah, right. So if we think about the spread of antibiotic resistance, right? And, this antibiotic resistance can arise in a couple of different ways. It can be through de novo mutations. So just like, a single bug within its genome acquires a mutation, say it mutates a target to a drug, and now it's resistant or tolerant. But another main route, another mechanism by which antibiotic resistance arises is through gene sharing, horizontal gene transfer.
Right.
And so those mechanisms of gene transfer, so there can be transduction, the movement by phages. There can be conjugation. So this is known as bacterial mating, where there's like this mating pilus, right? And then there's transformation, right? So bacteria taking up naked DNA, and there's a handful of new ones that are arising. Mm-hmm. These like things like vesiduction, where bacteria are blebbing off parts of their membrane, things like this. But what they all have in common is not just time, right? Things have to be right, not just somewhere at the same time, but they have to be in the same place. Once you have spatial structure, now you add gradients and you have basically spatial constraints. Like horizontal gene transfer is by nature going to be a spatially limited process. So, for example, a bug in the small intestine will have a harder time sharing genes with a bug in a colon. Maybe they are like ships passing the night at one point, they do pass each other, but it's probably more likely that co-inhabitants of certain regions of the gut or certain environments are going to share genes much more often, and that's what you see in genomes. And so when we think about space and time within the gut and seeing this and informing our idea about gene flow networks, space is undoubtedly going to have a role in either facilitating more transfer between organisms that are living next to each other, or maybe it constrains them because it essentially creates barriers to that transfer.
It's funny you'd say this because for many years I've been interested whether or not horizontal gene transfer can take place within marine filter feeders like mussels and oysters, which is one reason I have strong thoughts about consuming filter feeders.
Yes, there's shenanigans going on there too. Yeah. I mean, I think I think the rule would be that gene transfer is probably happening in every biological system, every niche. And even some of these filter feeders that might have the ability to destroy bacteria, maybe they're bacteriivores like, C. elegans, right? Once you crack open a bacterial cell, you're just taking all the recipe book that it has, right? Its genome, and you're just like throwing the pages out in the environment. And some other bugs, say like that are very good at taking up genes, like Neisseria and Strep, right? They're just like, thank you, thank you. And some Vibrios too, like Melanie Blockish's work with this amazing kind of system where the Vibrios are like taking in DNA, stuff like this.
So like Ariana Grande says, it's thank you next, right?
Yes.
And this is interesting because the thing that you said that really appealed to me is if you were just broadcasting DNA by cell lysis and it was in a large volume, very little opportunity for the other organisms to pick it up. But we don't think about what takes place in a biofilm, and there are pockets within biofilms, and that's really relevant. So your work is actually really applicable to a whole bunch of people.
100%. Yeah. No, it's very exciting. Yeah.
Do you have a particular observation you care to share with us about something that you found out that really, sounds like everything excites folks in your lab, but there you go.
Yeah, actually, there's two. If we have time to share them, I think the first one is the nature of a phage outbreak, right? So we work with temperate phages that are integrated inside of the bacterial cell. So we can colonize zebrafish with bacteria that are kind of already carrying their viral partner, their viral passenger. And these viruses that we work with, they become triggered to enter into lytic replication by stress. So if they sense their host cell undergoing stress, they kind of make a decision that they should kind of forego the vertical transmission that they get from bacteria growing and replicating to one where they jump ship essentially and infect a new host. So when we look at the spatial structure of bacteria within the gut, we actually see that it's highly spatially organized in some regions and in some bacteria. So some bacteria will form these clonal clusters within the gut. But then when we induce an outbreak, say with an antibiotic, and you get the phage replicating, what was really interesting is that we observed phage replicating kind of on the periphery of these aggregates. And when we watch this and we kind of track the dynamics of those phage leaving those populations and infecting another susceptible population, we haven't shown this, and this is where some of our experiments are going, but we have to imagine that this spatial structure is both creating the niches where the phages are even going to replicate, and then because of the spatial organization, it's going to limit, and in some cases facilitate, where those phages disperse and who they're going to infect. And so oftentimes when we kind of see kind of these large sequencing data sets on phages and we see they're there, we see they're replicating, but now we're like, where, when, who, like, how is it possible? And also when people say, oh, we didn't see much, well, maybe because there's a ton of spatial structure and it's completely landlocked. And so this is something that really has our gears going now after we've seen it. And so we have some plans of attack there.
It's interesting because when people hear about bacteriophages, and I only know this because I used to do some work with the bacterial predator Vibrio. And one of the things that when you're studying any plaque-forming bacteriophage, the plaque gets to be a certain size and no larger because generally speaking, when a bacterium stops growing, it's not that they become immune, but you no longer get phage replication. And this absolutely ties into what you just said. Areas of active growth, areas of quiescence.
Right.
It's so interesting.
That's right. And so if we think about something that's really compelling to me, if we go back to this idea of building microbiomes, what does that take? Well, I think we should be looking at the most ancient microbiome engineers of all, and that's phages. And so if we can think about how phages are naturally engineering microbiomes, I think we want to learn those tricks, those strategies for how they're spreading genetic material across a population.
That is amazing.
Yeah, and then I would say if we have time, the second thing that was really amazing, right? So we're talking about the interactions between phages and bacteria, right? So phages infect prokaryotic cells. And there's been research emerging over the last several years. Jeremy Barr is one of these people from Forrest Rohrer's lab originally that kind of, I think, at least for me, was my first kind of point of awareness for, hey, phages might not just be interacting with prokaryotes, they might be interacting with eukaryotes too. And so when we watch phage outbreaks within the gut, what really jumped out to us immediately, and when I say us, I mean it was Liz's sharp eyes that found this. The phages, the phages are interacting with the zebrafish gut tissues, and we have like amazing movies of this where we see phage particles, dancing around inside of the tissues. Like they're deep, they're many cell layers deep into the tissues. They're even inside of cells. And then what's amazing is that Liz noticed that the phages were being trafficked in the bloodstream and they are in the liver, right? They're somehow transiting from the gut into the bloodstream, into the liver. There's other labs that, June Round, Paul Boyke, who are studying phages and have found that, phages are interacting with the host immune system. And there's a lot of implications here, right? Because if you look at a phage and its components, right, it's foreign, right? It's a protein packaging, protein tail structure, and then they have this microbial nucleic acid inside. These are potentially inflammatory, maybe anti-inflammatory antigens, right? Stimulants for the immune system. And so we were just in awe of what we had just saw. We saw these viral particles now interacting with multiple cells and tissues and organ systems of this zebrafish animal. And it was really only made possible because this transparent vertebrate animal, we're able to zoom out to the whole body plan. And we're saying, oh my gosh, phages in the liver, phages in the blood, phages in the brain, phages in the, and so now we're just full of question marks and ideas, and hopefully, the future, hopefully 2025 is we have a lot of new questions for ourselves to ask.
It's interesting because when people learn about a field, they look for rules. And that's what they're all about, are the rules. Mm. And I'm always whenever I teach about microbiology, I was once critiqued that I gave a very general description of how peptidoglycan was put together, part of the cell wall of both gram-negative and gram-positive bacteria. But I didn't get terribly specific, and I was once critiqued by a non-microbiologist on this subject, and I said, because there's such variability, if I were to teach what E. coli does, and that's not true for all E. coli either, what's the point? So I stick with the basics of it. And then the rest is, the way I describe it to students is I describe the tree and then we put leaves on the tree. So I give them the trunks and the branches. That's my idea.
That's right.
But a physics TA put it to me many, many years ago, constants aren't and variables don't. And what I mean by that is that sometimes the rules that we've learned are simply not rules. And a good example is the idea, if you were to give a true or false question, bacteriophages interact with eukaryotic cells. Most people taking microbiology would say false, even though there's now evidence from several different groups, including your own.
Yeah, that's right.
And that's what's frustrating for students. Of course, I understand that they want there to be rules, but very exciting for me.
Yeah. Mark, 100% agree. I actually have a slide in my general microbiology class that says microbes are rule breakers. And I try to be very, very clear that my goal in a microbiology course is to give, as you say, the trunks and the branches, right? The basics, but just to prime them. And I tell them about the real experience I had as a microbiology student. At some point I got to grad school and I was like, hey, wait, the textbook is not even right, or it's not complete.
That's right.
But I think just like letting them know that like they need to be on the lookout for all these rule breakers. And when, and these are awesome opportunities. When we find a rule breaker, now we need to crack that open and ask more questions and ask if the original rules we had were just another game entirely, right? Based on another game entirely.
Well, if you look very carefully when you look at the history of microbiology, you look at like Pasteur and Koch and and they did wonderful stuff, but it was all based on isolation of a single organism, growth in single colonies, growth in pure culture. None of these things are relevant to the natural world. However, that doesn't mean what they did wasn't important. It was vitally important, but now we build on it to look at the vast and various microbial world around us.
That's right. Certainly open the door.
It just always makes me laugh when I see it in my own building. If you look around, you'll see it in yours, where someone will put a sign, "No bacteria in this room, please." And you're thinking, way too late.
Yeah.
First evolved, last extinct, right?
Yeah. I remember the first time in a pathogenesis lab where someone came in who worked on cancer, right? So they do tissue culture, but in a different context. And they noticed that we had bacteria all over our tissue culture room. And they're like, isn't that bad? It's like, that's how we do this experiment. We mix the kingdoms of life here. That's what we do.
No, it's so interesting to me. And it really is a different way of looking at the world. And there have been people who've written books on this subject. Ed Yong comes to mind. Really important stuff. But it's something that we really need to start hammering home as early as possible. Yeah. And I'm glad to hear the things that you're talking about today.
Yeah, well, I mean, maybe something I'll share, an anecdote. I had a chance that as a postdoc conversation with Scott Gilbert, who literally wrote the book on evolutionary developmental biology. And he really captivated me with some of his writings on the holobiont. And I had a chance to have a conversation with him. I was like walking him between meetings. And we were having a really animated conversation. I really dig a lot of the thinking and philosophy behind some of the writings he's had in the recent years. But he stopped. I remember like stopping on the street and he said, you have to remember in life, teams win, not individuals, teams. And what he's referring to there, and something I share with my class that, like, to your point, we got to like start this sooner. You have to understand that like we are a collective, right? We are a composite of all these. And so then when we kind of look at phages transiting into the, right, we have this inter-kingdom relationship interactions going on. This is the rule of life. It is a mixed up kind of messy business biology is. And so the quicker we understand that the barriers are not as strict, and this goes, into the disciplines that we study, not just the biological systems. I think this is something I often try to iterate across much quicker when we're thinking about hypotheses and mechanisms in my lab, is how to flip it on its head to say, well, what if the rules are wrong? What if we need to think more fluid, more porous about this problem? This is a really exciting area, I think.
So I don't want to take up more time than I have to with this, but when I was in college, I was very interested in Zen Buddhism. And there's this lovely idea called the beginner's mind. I will soon add it to my tattoo collection, by the way. But the fact of the matter is you need someone with a beginner's mind. And that doesn't mean dumb. That doesn't mean unknowledgeable. It means someone who doesn't have preconceived notion. They're open to change. And that is the one thing I've learned in the microbial sciences, is that it's about change. And it shouldn't surprise anyone, because the microbial world, again, first evolved, last extinct.
Yeah, 100% agree.
So do you have a last bit of wisdom to share with our listeners and viewers?
Well, I think what some of the topics we've been talking about have really hit on this. I think if I were to think about some wisdom, I would say for your novice micronauts, I would say get unsettled about something, right? And follow that feeling. When I started out, I didn't know I'd be studying what I'm studying now, but I just knew that I was unsettled. I was uncomfortable not knowing something, and I just kept scratching at it, right? And I just kept going and asking more questions, and I just knew that I liked that. I found a lot of satisfaction in that. So I think for your novice micronauts, just follow that feeling. Don't hide from it. Don't run from it. There's like so much we try to chase comfort. Do your best to get comfortable being uncomfortable, right?
Yes.
And as you keep following that, I think you'll start getting into things that you find intriguing and engaging. And I think that brings, satisfaction and happiness. So I would just say that would be my wisdom and advice. And of course, that's informed by my scientific trajectory, being faced with challenges and kind of seeing things we didn't even think we were going to see. But I think that scales up to just how we approach life in general. So chase those unsettled feelings. Don't run from them.
The three most important words in science are "I don't know." And students hate it, but it's really an opportunity.
Yeah.
And that's one thing that has really informed my entire career, is I want to know more.
Yes. Yes.
And
100%.
and listening to you, I certainly want to know more.
Yeah. Awesome.
So I want to thank you for your time today, and I want to wish you and your lab group the very best of luck. I have family in Irvine, so I'm going to have to make an excuse to come visit. Maybe
Yes,
I can look through
absolutely.
one of your
Absolutely.
microscopes too.
Yes. Awesome. It'd be super fun. Super fun. Thanks for having me, Mark. This has been awesome.
It's been great. Thank you. This has been Matters Microbial, a weekly podcast about the wonder of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode with epic links can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Travis Wiles is Assistant Professor of Molecular Biology and Biochemistry at the University of California, Irvine. Many thanks to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our quality quorum today. See you next time on Matters Microbial.